As the year comes to an end, we also hope that this is the beginning of the end of the COVID-19 pandemic that almost threatened the very existence of humanity. As countries across Europe and the United States of America start rolling out vaccinations, we at Treat by Alterdoctors present this very special episode, Watchdog, to look at the data and the science behind the vaccination. Over the years, there have been multiple types of vaccines, from live attenuated and killed vaccines to toxoid or subunit vaccines, and today we are in the era of genome-based approaches for vaccine. But let's understand the terminology which has been floating around for the last few weeks. What are mRNA vaccines? But before that, let us do a quick refresher course in biology to figure out what exactly is mRNA. So we all know that the genetic information and instruction is stored as DNA within the nucleus of the cell. But how does this instruction get relayed to the cell to carry out its activities? Very simply through the messenger RNA or the mRNA. So the information in the DNA is transcripted to mRNA, which acts as a template and instructs the ribosomes to synthesize proteins which are necessary for the cell to perform its activities. But why are we talking about mRNA when it comes to the COVID-19 virus? It's simple. SARS-CoV-2 belongs to the coronavirus family, which is a RNA virus. Now, the RNA genome in the virus codes for multiple proteins, one of it being the spike protein, which is expressed on the surface of the virus, which helps it in recognition and attachment of the virus to the cells in the human body. So, how did the mRNA vaccine develop? So initially, scientists focused on understanding the sequence in the RNA genome of the virus that coded for the spike protein. Once that was accomplished, they synthesized the mRNA sequence, which when it would be delivered to the human cell would make the same spike protein. The mRNA was packaged in a lipid nanoparticle which served as a delivery vehicle to carry the sequence to the cell. So essentially, the mRNA sequence along with the lipid nanoparticle became the vaccine which would be injected intramuscularly. Once inside the cell in the human body, the mRNA issued instructions for the cell to make the spike protein and display it on its surface. Once the protein was synthesized, the mRNA was degraded. Now, when the body's immune cells come in contact with these cells expressing the spike protein, they recognize it as a foreign object. Thus, the immune system gets activated, leading to recruitment of T cells and formation of antibodies. So, what are the learning points from this theoretical aspect of mRNA vaccine? There is no coronavirus or COVID-19 virus in the vaccine. It is a synthetic genetic material, mRNA sequence, which gives instructions to the cells to manufacture and express the spike protein, which is a viral protein expressed by COVID-19 during an actual infection to stimulate the immune system. However, because it is expressing only the viral protein and there is no involvement of the entire viral structure, there is no risk of infection because of the mRNA vaccine. The advantage of this? The immune system of the body is able to generate a protective immune response by producing antibodies without an actual infection occurring. Therefore, when exposure to COVID-19 occurs, the body already has antibodies which makes it capable of fighting the virus. Furthermore, the mRNA sequence gets degraded once the protein is expressed and does not last in the body forever. All this sounds fantastic in theory, but how did it fare in the clinical trials, which is ultimately the proof of the pudding? We have the safety and efficacy of the BNT162b2 mRNA COVID-19 vaccine. Now, I've chosen this vaccine because the full details of the phase 2/3 trial have been published in a peer-reviewed journal. So let us take a look at how well did it perform and what were the safety events. So it was a phase 2/3 trial. 21,720 people received the vaccine and 21,728 received the saline placebo, meaning they did not receive the vaccine but they just received a shot of normal saline. So who were the participants in the study? So essentially adults who were 16 years of age or older who were healthy or had stable chronic medical conditions were randomized to one of the two arms of the study. Anyone with a past history of COVID-19 or were on immunosuppressive therapy or were immunosuppressed were excluded from the study. The study, however, did include patients with stable HIV disease, hepatitis B and hepatitis C. Those in the vaccine arm received 2 doses of the vaccine, which was administered intramuscularly 3 weeks apart. And basically they looked at the efficacy and the safety of the vaccine. So efficacy was defined by how many developed confirmed COVID-19 among those who received the vaccine, with the onset being at least 7 days after having received the second dose of the vaccine as compared to those who did not receive the vaccine. They also looked at the safety profile by looking at how many of those who received the vaccine developed either local or systemic adverse events. Now coming to the baseline demographic characteristics, there were 18,860 participants in the vaccine arm and 18,846 in the placebo arm. 58% in both the arms belong to the 16 to 55 years age group, while 42% belong to more than 55 years age group. So by observing how many developed COVID-19 with onset at least 7 days after the second dose, we can calculate the efficacy. Now among the 18,198 who received the vaccine, 8 developed COVID-19 as compared to 162 participants who developed COVID-19 among those who did not receive the vaccine. This simply translated to a 95% efficacy for the vaccine, which starts 1 week after the second dose. But now questions remain. Are 2 doses really required? Is 1 dose not enough? So if we look at the trial data a little deeper, we will have the answer to this question also. So between the first dose of the vaccine and the second dose of the vaccine, in the vaccine arm, 39 developed COVID-19 as compared to 82 who developed it in the placebo arm, which did not receive the vaccine. Now this translates to an efficacy of 52% for the vaccine, meaning to say that after 1 dose, there is some amount of protection being offered, but the best protection and the best efficacy comes in after 2 doses of the vaccine. A lot of important questions remain about safety and what did the trial tell us about the local and systemic adverse events. So with a median follow-up of 2 months, an analysis of the safety data subset showed that the most common local adverse event was local pain at the injection site, which was mainly mild to moderate in nature and resolved within 1 to 2 days. The most common systemic adverse events which was reported across age groups among those who received the vaccine was fatigue and headache. Now looking at the data that we have at this point of time, with a median follow-up of 2 months, the vaccine does appear to be considerably safe. Also, the amount of serious adverse events which happened, though it occurred in a very minimal number of participants, was similar both in the vaccine arm and in the placebo arm. So let us not go by false media reports, let us not go by rumors or any hype. At this point of time with this follow-up, the vaccine does appear to be considerably safe. Of course, there are a lot of further questions to be answered. A longer follow-up, more clinical trials and further data sets should help us to answer these questions in the near future. But with multiple vaccines being available, how would governments choose the vaccine? Well, I think it would depend on a multiple of factors such as the storage required, the number of doses required, the cost involved, apart from the scientific data of efficacy and safety. With vaccines now being rolled out in many countries, there is a glimmer of hope that the pandemic may soon be making its way towards its end. Thank you so much for watching this special episode of Watchdog from Treat by Alterdoctors. You can visit our website www.alterdoctor.in. You can also follow us on our social media handles. If you have any articles, art, photography or any creative piece that you would want us to publish, please do send it to us at treatfordoctors@gmail.com. This is your host Dr.Nitin Yashas on behalf of Treat by Alterdoctors, wishing you a very Merry Christmas and a happy, healthy New Year ahead. Thank you.